Pteridophytes include horses, ferns. Pteridophytes are used for medicinal purposes and are soil binders. They also frequently grow as ornamentals. Evolutionary, they were the first terrestrial plants to possess vascular tissues like xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish in sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is gametophytic. So in bryophyte, dominant phase is gametophytic plant's body. However, in pteridophyte, the main plant body is sporophyte and is differentiated into true root, true stem and true leaves, unlike the bryophytes wherein false roots, stem and leaves were there, or root-like, stem-like and leaf-like structures were there. These organs possess well differentiated vascular tissues. The leaves in pteridophytes are small. Leaves are small, therefore they are called microphylls, as in Selaginella, or large, that is macrophylls, as in ferns. The sporophyte, which is the main plant body, sporophyte bear sporangia that are subtended by leaf-like appendages known as sporophyll. In some cases, sporophyll may form distinct compact structures called strobili or cones. Example, Selaginella and equisetum. Strobili and cones, Selaginella, equisetum. The sporangia produces spores by meiosis in the spore mother cells. Spores germinate to give rise to inconspicuous, small but multicellular free-living, mostly photosynthetic thalloid gametophyte called prothallus. These gametophyte require cool, damp, shady place to grow. Because of this specific restricted requirement, the need for water for fertilization, the spread of living pteridophyte is limited and restricted to a narrow geographical region. The gametophyte bear male and female sex organ and theridia and archegonia respectively. Water is required to transfer antherozoids. Male gametes are released from antheridia to the mouth of archegonium. Fusion of male gamete with egg in the archegonium result in formation of zygote. Zygote thereafter produces a multicellular, well differentiated sporophyte, which is the dominant phase in pteridophyte. In majority of pteridophyte, all spores are of similar kind, homosporous, like Selaginella salvinia. Selaginella and salvinia produces two kinds of spores: macro, which is large, micro, which is small spores, known as heterosporous. Megaspores and microspores germinate to give rise to female and male gametophyte. respectively. Female gametophyte The female gametophyte in these plants are retained on parent sporophyte for variable periods. The development of zygote into young embryos takes place in female gametophyte. This event is a precursor to the seed habit, considered as the important step 2 in evolution. Pteridophytes are further classified into 4 classes: Xylotum, Xylopsida, Xylotum, Lycopsida, Selaginella, Lycopodium, Spanopsida, Equisetum, Pteropsida, Dryopetris, Petris, Ediantum. So this is what pteridophytes are about. Let us look at the diagrams. Pteridophytes, they have Selaginella, they have Equisetum, they have fern, they have Salvinia. Selaginella are the ones that have microphylls in Selaginella and macrophyll in ferns. The Selaginella has microphylls and ferns have macrophylls. So this is Selaginella, this is fern. Another thing, Selaginella and Equisetum, they have strobili and cones. So this is Selaginella, it has strobili and cones, and strobilus is clearly seen in Equisetum. This is a node, this is an internode, and these are branches, and this is rhizome. Then heterosporous are Selaginella and Salvinia. So Selaginella and Salvinia are heterosporous. They produce male and female organs or sex organs of different size. Heterosporous, the megaspores they produce two kinds of spores, macrospores and microspores. Megaspores and microspores germinate to female and male gametophyte respectively. Selaginella, Equisetum, fern, Salvinia. Gymnosperms are the plants in which ovules are not enclosed by ovary wall. They remain exposed both before and after fertilization. Seed that develops post fertilization are not covered and are naked. Gymnosperms include mid-sized trees, tall trees and shrubs. One of the gymnosperm, giant redwood tree sequoia, is one of the tallest trees species. The roots are generally taproot. Roots in some genera have fungal association to form mycorrhiza, that is with Pinus. In some cycas, small specialized roots called coloroid roots are associated with nitrogen fixing cyanobacteria. The stems are unbranched cycas or branched Pinus or Cedrus. So Pinus has Pinus has mycorrhiza as association, and Cycas has caloroid roots as association. Nitrogen fixing cyanobacteria, caloroid roots they are associated with nitrogen fixing cyanobacteria. So C for Cycas, C for caloroid root, and C for nitrogen fixing cyanobacteria. Cycas is more like an inverted U. The C is more like an inverted U, so you can see there's no branch, whereas when it comes to Pinus, it has branch, therefore it is branched, along with Cedrus. Cedrus is also branched. The leaves may be simple or compound. In Cycas, pinnate leaves are persistent for a few years. So leaves could be simple, leaves could be compound, and in Cycas, which is unbranched, pinnate leaves persist for a few years. Leaves in gymnosperms are well adapted to withstand extreme temperature, humidity, and wind. Conifers, the needle-like leaves reduces the surface area. Thick cuticle, sunken stomata reduces water loss. Gymnosperms are heterosporous. They produce haploid microspore and megaspore. Two kinds of spores are produced within the spongia that are borne on sporophylls, which are arranged spirally along an axis to form lax or compact strobili or cones. So gymnosperms are heterosporous. Two different types of spores are produced, and that too haploid in nature. And both of them are formed within the sporangia. Sporangia is borne upon sporophylls, and sporophylls are radially arranged in a radial manner, and that's how they form lacs or strobilus or cones. The strobilus bearing microsporophylls and microsporangia are called microsporangiate. So microsporangiate or male strobili. The microspore develop into male gametophytic generation, which is highly reduced and is confined to only a limited number of cells. The microspores develop into male gametophytic generation, which is highly reduced and confined to only limited number of cells. This reduced gametophyte is called pollen grains. This reduced gametophyte is called pollen grains. This reduced gametophyte is called pollen grains, which is only limited to number of cells. The development of pollen grains takes place within the microsporangia. The cones bearing megasporophyll with ovule or megasporangia are called megasporangiate or female strobili. Male and female cones or strobili may be borne on the same tree, Pinus, because Pinus, if we look at it, is branched. So therefore it will be easy for us to It will be easy for the pinus to have both of them on the same tree. Whereas in cycas, because it is unbranched, therefore the male cone are born on different trees. Megaspore mother cell differentiated from one of the cells of the nucleus. The megaspore mother cell is differentiated from one of the cell of nucellus. Nucellus is protected by envelopes, and a composite structure is called an ovule. So megaspore mother cell differentiated from one of the cells of nucellus, and the nucellus is protected by envelopes, and the composite structure is called ovule. Ovule are born on megasporophyll, which may be clustered to form female cones. The megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspore, so the megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspores enclosed within the microsporangium develop into multicellular female gametophyte. Female gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained on megasporangium. Unlike bryophyte and pteridophyte, in gymnosperms, male and female gametophyte do not have independent free-living existence, which means unlike bryophyte and pteridophyte, in gymnosperm, male and female gametophyte do not have independent free-living existence, but they remain within the spongia retained on the sporophyte. The pollen grains are released from the microsporangium. They are carried in air currents and come in contact with opening of the ovule borne on megasporophylls. The pollen tube carrying male gametes grow towards the archegonia in the ovules and discharge the content near the mouth of the archegonia. Following fertilization, zygote develops into embryo, ovule develops into seed, and these seeds are not covered. So the pollen grain are released from the microsporangium. They are carried by air currents. They come in contact with the opening of the ovule on the megasporophyll. The pollen tube carrying male gamete grow towards the archegonia in the ovule, discharge their contents near the mouth of archegonia. Following fertilization, zygote develops into embryo and ovule into seeds, and these seeds are not covered. This is cycas, which is unbranched, and it has male and female gametophyte on different plants. Pinus, which is branched, it has male and female gametes on the same plant. This is ginkgo. Ginkgo has a dwarf shoot, long shoot, and seeds. Angiosperms. Unlike gymnosperms, where ovules are naked, in gymnosperms or flowering plants, Pollen grains and ovules are developed in specialized structures of the flowers. In angiosperm, seeds are enclosed in the fruits. Angiosperms are an exceptionally large group of plants occurring in wide range of habitats. They range in size from smallest Wolffia to tall trees eucalyptus over 100 meters. They provide food, fodder, fuel, medicines and several other commercially important products. They are divided into two classes, dicotyledonous and monocotyledonous. Dicotyledonous characterized by seeds having two cotyledons, reticulate venation, tetramerous and pentamerous flowers, which is having 4 or 5 members of each floral wall. Monocotyledonous, on the other hand, has single cotyledon, parallel venation, trimerous flowers, that is having 3 members of each floral whorls. Male sex organ in flower is stamen. Each stamen consists of slender filament with an anther at the tip. Within the anther, pollen mother cells divide by meiosis to produce microspores, which matures into pollen grains. The female sex organs in a flower is pistil, and pistil consists of a swollen ovary at its base. A long slender style stigma inside the ovary. Ovule is also present. Generally, each ovule has megaspore mother cells that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and 1 form the embryo sac. Each embryo sac has 3 egg apparatus, 1 egg cell, 2 synergid, 3 antipodal, 2 polar nuclei. Dicotyledonous, monocotyledonous. Nuclei eventually fuse to form diploid secondary nucleus. Pollen grains, after dispersal, from anther, carried by winds or various agencies to the stigma of the pistil. This is termed as pollination. The pollen grains germinate on stigma and, resulting in pollen tube, grow through tissues of stigma and style and reach the ovule. The pollen tube enters the embryo sac where 2 male gametes are discharged. One of the male gametes fuses with the egg cell, syngamy, to form a zygote. Other male gamete fuses with diploid secondary nucleus and produces triploid endosperm nucleus pen. Because of the occurrence of two fusions, that is syngamy and triple fusion, 2 types of fusion, that is syngamy and triple fusion. This event is known as double fertilization, unique to angiosperms. The zygote develops into an embryo which has 1 or 2 cotyledon. A PEN develops into endosperm, which provides nourishment to the developing embryo. The ovule develop into seed and ovaries develop into fruit. The life cycle of angiosperms shown in the figure 3.6. Sporophyte, megasporangium, ovary, style, stigma. Inside ovary, we have megaspore, mother cells. Then we have male gametophyte, which is anther and filament, which forms microsporangium. Megaspore mother cell further divides to form egg. Microsporangium further divides to form microspores. Then pollen grain, which is also a microspore. Then the male gametophyte. Two of the pollen grains or gametes are released to the egg and fusion occurs, and that's how the zygote is formed. Then the embryo is formed. That's how the sporophyte is formed. This is what a life cycle of angiosperm looks like. Plant life cycles and alteration of generation. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. This in turn divides by mitosis to form haploid plant body once again. Thus, during the life cycle of sexually reproducing plants, there is an alteration of generation between gamete producing haploid gametophyte and the spore producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in following patterns. Sporophytic generation is represented only by one-celled zygote. There is no free-living sporophyte. Meiosis in the zygote results in formation of haploid spores. The haploid spores divide mitotically to form the gametophyte. The dominant photosynthetic phase in such plant is the free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as volvox, Spirogyra and some species of Chlamydomonas represent this pattern. On the other extreme is the type where the diploid sporophyte is dominant. Photosynthetic independent phase of plant. The gametophytic phase is represented by single to few-celled haploid gametophyte. This kind of life cycle is termed as diplontic. An alga, fucus represent this pattern. In addition, all seed-bearing plants, that is gymnosperms or angiosperms, follow this pattern in some variation wherein the gametophytic phase is a few to multicelled. Bryophyte and pteridophyte interestingly exhibit an intermediate condition, haplodiplontic, because both the phases are multicellular. However, they differ in their dominant phase. A dominant independent photosynthetic thalloid or erect phase is represented by haploid gametophyte, and it alternates with short-lived multicellular sporophyte totally or partially dependent on the gametophyte for its encourage nutrition. All bryophyte represent this pattern. The diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. It alternates with the multicellular sporophytic and autotrophic independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophyte exhibit this pattern. Interestingly, while most of the alga generation is haplontic, some of them such as Ectocarpus, Polysiphonia, Kelps and are haplodiplontic. Fucus and alga are diplontic. Summary. Plant kingdom include algae, bryophyte, pteridophyte, gymnosperm, and angiosperm. Algae are chlorophyll-bearing, simple, thyroid, autotrophic, largely aquatic organism. Depending on the type of pigment possessed and the type of stored food, algae are classified into three classes, namely chlorophyceae, phaeophyceae, rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different types of spores, and sexually by formation of gametes which show isogamy and isogamy-oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of alga. It is thallus-like and prostrate or erect and are attached to the substratum by rhizoids. They possess root-like, leaf-like, stem-like structures. Bryophytes are divided into liverworts and mosses. The plant body of liverwort is thyloid, dorsiventral, whereas mosses are upright. Slender axis bear spirally arranged leaves. The main plant body of bryophyte is gamete producing and is called gametophyte. It bears male sex organs called antheridia and female sex organs called archegonia. The male and female gametes produced fuse to form zygote, which produces a multicellular body called sporophyte. This produces haploid spores. The spores germinate to form gametophytes. In pteridophyte, the main plant body of sporophyte is differentiated into true root, stem, and leaves. These organs possesses well differentiated vascular tissues. The sporophyte bearing sporangia which produce spores. The spore germinate to form gametophyte which require cool damp places to grow. The gametophyte bear male and female sex organ called antheridia and archegonia respectively. Water is required for transfer of male gamete to archegonia, where the zygote is formed after fertilization. The zygote produces a sporophyte.